Okay, welcome and thank you for joining the SADS Foundation webinar today, September 22nd, 2025. I'm Marcia Baker, the SADS Foundation's Program Director. This webinar will be an overview of Solid Biosciences gene therapy clinical trial called ARTEMIS for CPVT, or catecholaminergic polymorphic ventricular tachycardia. On behalf of the SADS Foundation, we thank Solid Biosciences for its grant support to make this webinar possible. Now, I'd like to introduce our presenters today. Dr. Gabriel Brooks is the Chief Medical Officer of Solid Biosciences and leads the clinical function of all Solid's neuromuscular and cardiac programs. And Annie Ganot, who's Senior Vice President and Head of Patient Advocacy and a co-founder of Solid Biosciences. They will introduce you to Solid Biosciences as a company and provide an overview of their gene therapy trial for people living with CPVT and how a person can participate if interested and eligible. Following the presentation, you'll have the opportunity to ask your questions by posting your question in the chat, and the questions will be addressed at the end of the presentation. A reminder that this webinar is being recorded, so please keep your microphones and audio muted during the presentation. The webinar recording link will be emailed to all of you in a few days. So welcome, Dr. Brooks and Annie.
That's great, Marcia. Thank you so much for that introduction. I'm just going to share my screen and bring up our slides. Wonderful. Can you all see the slides okay?
Yes.
Fantastic. I'm just going to go ahead and advance. So I can even just skip this slide since you did such a beautiful job introducing us. Thank you so much. We are grateful to SADS Foundation for hosting this webinar, and of course to the CPVT community for taking the time to be here. Again, I'm Annie Ganot. I'm one of the co-founders of Solid, and I lead patient advocacy. And I am joined by my colleague, Dr. Gabriel Brooks, who is our Chief Medical Officer. I also want to give special thanks to the SADS CPVT Steering Committee for being such a thoughtful partner and for sharing feedback and insights that have directly shaped the design of our clinical trial. Before we get started, I want to remind everyone that we are a publicly traded company. Today's discussion will include forward-looking statements. These are based on current expectations and assumptions and are subject to risks and uncertainties. So here's what we're going to cover today. We're going to provide a background on Solid Biosciences, how we began, what motivates our work, and how we've expanded our focus to rare neuromuscular and cardiac conditions. We will then provide context on how gene therapy works and why we believe it holds potential for CPVT. Then we will go into more detail on SGT-501 and the clinical trial overview, and of course, our study design, and also including preclinical data. We want to explain a lot about the ARTEMIS study so that we can try to address any questions that you may have. And of course, at the very end, we will leave it open to time to take questions from the patient community. So thank you so much. We hope this session provides a clear picture of where we are in our journey, why we're excited about the potential of this program, and how grateful we are to be working side by side with the SADS Foundation and the CPVT community. So just a little bit of background on Solid Biosciences. We were founded in 2013 after my son was diagnosed with Duchenne muscular dystrophy. What began as a deeply personal mission has grown into a precision genetic medicines company dedicated to developing meaningful therapies for patients living with serious neuromuscular and cardiac conditions. Over the past decade, we've built expertise in gene therapy and patient-centered drug development. While our initial focus was Duchenne, we have since expanded our portfolio to include Friedreich's ataxia and rare cardiac indications such as CPVT. We are headquartered in Boston, Massachusetts, and our team is united by a single purpose: to bring life-changing therapies to patients and families who urgently need them. Everything we do is driven by science, but also by the voices and experiences of the patient communities we serve. With that, I'm going to turn it over to my colleague, Dr. Gabriel Brooks.
Thank you so much, Annie. And let me take this opportunity to thank Marcia and the SADS Foundation leadership, Dr. Sylvia Priori and her lab, who's really forms the basis of what we hope is a transformational drug for the CPVT community. But most importantly, my thanks are to each and every person dialing in, because patients and their families, yes, we are developing therapies we hope to address their disease, but it's only made possible by partnership with patients who join us as participants. So that's critically important for all of the work we do. So we're going to start at a very basic level, since we are talking about a new medicine that is a gene therapy. So first we start with, what is a gene? And a gene is a chemical that's part of our body, but an amazing feat of nature is this chemical actually contains the instructions for every cell and every protein in our body. And this chemical is called DNA, which is translated into a message, RNA, and then that message is translated into a protein. And in this case, we're talking about RYR2, the ryanodine receptor protein that is abnormal, it being treated with a calsequestrin. So the DNA is the gene for calsequestrin, which is translated into the RNA of calsequestrin, and that is developed into the protein Cas-Q2. So what is gene therapy? If that's a gene, then what is gene therapy? So we have a new gene, the one that we are the therapeutic gene. In this case, it's Cas-Q2. And in the context of a gene that's not working well, and in this case, we're primarily focused on the ryanodine receptor, the RYR2 gene. We're talking about gene replacement therapy. So in this case, we're expressing more of this Cas-Q2 gene to try to drug, and we'll walk through this, the malfunctioning RYR2 gene. This is different than gene editing. In that case, that is actually transforming a gene in your body and changing that gene. This is what this therapy is gene replacement therapy as opposed to a gene editing therapy. So in terms of how we actually make this therapy work, what is needed for a gene replacement therapy, the first thing you need is you need a way of delivering your gene. And in this case, it is the calsequestrin gene, again, to make function more normally that abnormal ryanodine receptor gene. And how we get that into the nucleus of the cell, that chemical that is the instructions for making calsequestrin, we use viral capsids. So what is a viral capsid? You can see a picture of it here. It looks like a 20-sided die, but each one of the proteins on that capsid allows for the delivery from the outside of the cell through the cell and into the nucleus of that gene. And that is something that has been created over the billions of years that life has been on this planet, is the ability for viruses to deliver genetic material to the nucleus. So what is needed for gene replacement therapy really is to harness the power of how viruses normally work. Once that gene is into the nucleus of the cell, we then need a promoter to promote the expression of the gene that is being delivered. And then we need the gene itself. And in this case, again, it's that Cas-Q2 gene. We use as that capsid an AAV. So what is AAV? It's adeno-associated virus, which is expressed which we find in nature. It does not replicate on its own. It requires an adenovirus to replicate, and that's why it's called adeno-associated virus. What we have done is we've actually taken out all of the genes that are inside that adeno-associated virus and replaced it with this promoter and transgene, what we call a construct. So if we go to the next slide of how gene therapy works, this is a depiction of what I described. And again, you have this AAV capsid, which looks like a 20-sided die, that then goes into the cell. The proteins of the capsid itself have the instructions to deliver into the nucleus of the cell. You can see that as in the center column, that gray circle. That's our characterization of the nucleus, the genetic material that's being held within the virus. And of course, here we've taken out what naturally would be in an adeno-associated virus and put the genetic material of the calsequestrin gene. And then what happens is that that genetic material, that DNA, that calsequestrin gene, then produces protein using the normal function of the cell. So now that we understand what gene therapy is, a way to get our gene of interest into the cell and produce a protein using the cell's own machinery, now we can start to think about how is this different from traditional drug therapy. So traditionally, we give pills or other medicines to manage a disease by alleviating its symptoms, maybe on target, but a lot of times it is the effect of the disease that we're trying to mitigate. Whereas with gene therapy, the goal is to target the specific cause of the disease itself. So if you will, upstream, where we actually see, if we go back one slide, yeah, where we actually are seeing the biology that is abnormal. A traditional drug would require a dose every day. Whereas with gene therapy, because we are supplying the body with the instructions to make that protein, only one dose is required. In terms of how long will this last? Well, a traditional drug will wash out of our systems and we'll need to take more. The goal of a gene therapy is that we believe it is likely permanent, but it's also non-reversible. So once that gene is in the nucleus, that's where it will stay. In terms of how we look at testing the safety and efficacy of new drugs, in traditional drug therapy, that can take months to years. Here, we absolutely need to look at least for 5 years over long-term follow-up because this type of technology is rather new. And we want to make sure that we're looking at the long-term safety effects of the drug. So as a participant with us, we'll be together for at least 5 years while we're watching to make sure that we can look at any safety or efficacy signal that may be occurring. What would the impact on you on a participant be if they did join this trial? And we'll be talking about some of the known observed risks with AAV gene therapies. One of the impacts it's important to recognize is that right now, with our current technology, it's likely that if you participate in a trial and you're exposed to a gene therapy, it's unlikely that you would be able to be a candidate for another gene therapy. So what are the risks associated with viral vector gene replacement therapies? So one thing that we have observed is that viruses, although the particular capsid, remember it's that shell, goes very well to the heart. We do know that viruses oftentimes go to the liver, and there have been observed liver injury. We also know that the viruses, just like viruses that we are exposed to in life, can cause immune reactions. We have seen, in very rare instances, heart inflammation. And we know that kind of one thing that I had touched on before, if you expose patients to these gene therapies, we know that the patients create antibodies that now prevent redosing, meaning effective exposure subsequently. So how do we think about mitigating those potential risks? Well, we use specific and selective immune suppression. We follow patients very closely in terms of making sure that clinically, physically, they're doing well, and we test their blood to make sure that their normal function is being maintained. We follow patients for that 5 years to make sure that they continue to be safe. And then in terms of making sure that the specific participant has the best chance of potential efficacy, meaning a potential good effect from the drug, we make sure to select our participants very carefully. We can further reduce risks from these therapies by being very careful in terms of the dose that we select. And that's something that we've considered very closely about Artemis, is to find the dose that we believe would be compelling for a potential therapeutic effect, meaning that has the highest chance of giving a therapeutic effect, but also minimizes the risk. Because one thing we know is that if we can reduce the dose, that likely means that the risk of having any of these potential risks could be even lower. So why therapy for CPVT? One of the critical pieces here is there's a convergence between our understanding of the disease and the technology that we have that opens the possibility, we believe, to precisely treating that pathophysiology, that understanding of what the disease is caused by. We also understand that the current therapies for CPVT don't treat the root cause of the disease, and so that gives us opportunities to more selectively, and we hope more potently, treat the underlying disease biology. We know that current therapies for CPVT can cause significant side effects, which can make the medicines that we take make us feel sick, and we hope to avoid that, obviously, but also may sometimes lead patients to not take the medicines, and it's in those instances that they can be at the highest risk for the disease manifestation. And we know, of course, with CPVT, the disease manifestation can be an unstable arrhythmia, which could be very dangerous to their health. So how does SGT-501 provide a potential gene therapy for patients with CPVT? So we understand that the RYR2 channel in CPVT1, the most dominant type of CPVT, has increased sensitivity to calcium that can allow calcium to go through in diastole. Diastole is the time in the heart cycle that is mostly quiet, and you don't want the heart to contract or to conduct an electric signal. But in CPVT, this extra sensitivity means that when patients are under stress, whether that's physical or emotional, that can increase our calcium loads. And even in that time when the heart should be quiet, you can have that abnormal flow of calcium, and that can trigger a very dangerous arrhythmia. So what are the ways that we are thinking of being able to address that is to add calsequestrin. And that extra calsequestrin can soak up the calcium and then normalize, we believe, the RYR2 channel's sensitivity so that even under stress, whether again that's physical or emotional, that RYR2 channel will be less leaky in that quiet period, the diastole. So even though we understand with CPVT1 that the abnormality is this extra sensitivity of the RYR2 channel, we believe that by providing the body with extra calsequestrin, that can reduce that sensitivity by soaking up that extra calcium and then making sure that the patients are not, we would hope, susceptible to that arrhythmia during stress. So then what are the components of solids gene therapy for CPVT? So number one, we're using an AAV capsid to target the heart cells. We're using a promoter. In this case, it's called a Desmond promoter, which means that the transgene here, CASQ2, is expressed in muscle. So we have something that's the capsid getting the gene to the heart. Now it's inside the heart nucleus, the cardiomyocyte nucleus. We have a Desmond promoter that says, okay, I'm going to express this gene because I'm in a muscle cell. And what is it expressing? It's expressing this calsequestrin Cas-Q2 transgene. And remember, the whole point of that is so that we can normalize the sensitivity of RYR2. And we can certainly go into a lot more of this during our question and answer period. So clinical trials support the development of new therapies like the ones we're working on, really in large part to research that's done at academic centers. And here is a picture of the Sylvia Priori lab, who really were able to, one, elaborate the disease pathophysiology, meaning teaching us all why we have these abnormal heart rhythms in CPVT, what is the mechanism of disease. And then they developed this approach of normalizing the RYR2 channel and diastole, giving us the prospect, we hope, to bring to the CPVT community what we hope will be a potent and safe therapy. So SGT-501 has been evaluated for treatment of CPVT in preclinical studies. So we've done numerous mice and large animal, here monkey studies, to look at the safety of the CPVT product, which is SGT-501, as well as in specific models where we can recapitulate the disease, meaning we can make the disease manifest by manipulating the genes in those animal models to be a CPVT phenotype, meaning looking like CPVT. We can test whether or not SGT-501, this therapy for CPVT, actually corrects the disease. And I'm really proud to share with you that indeed our data showed that, number one, we were able to dose safely at levels higher than were coming into the clinic, non-human primates. And two, when we look at that dose, it was certainly able to change the disease phenotype in those models, those animal models of disease. So now I'd like to go into the ARTEMIS study. I'll give you an overview. I'll share with you what we call the inclusion-exclusion criteria, but really what it is who are the participants that we would like to invite into the study, what it would look like to be a participant in the study, and what that would mean for the participant being in this study. So in terms of a study overview, ARTEMIS is what we call a phase 1b trial, which primarily focuses on safety and tolerability of SGT-501. This really means that we're looking very closely to make sure that we understand how SGT-501 works for the participant. And that means, yes, we're going to be looking at the arrhythmias. We're going to be following those patients to make sure that under physical stress that we hope will have reduced arrhythmia, but also we'll be looking at markers in their blood and other physical exams to make sure that that participant, we hope, will be doing well. The study is going to begin with adults, age 18 or older, and then if we can show that the participant those adult participants are doing well, we can then open it up to participants that are younger. Participants can be patients with or without implantable cardioverter defibrillators, or ICDs. Of course, all participants will have to have a clinical diagnosis of CPVT, but then we also will make sure that the gene that is causing their CPVT is due to what we call a pathologic or likely pathologic mutation in RYR2, meaning that the mutation that that particular participant has a high probability of being the thing that is causing their CPVT. All patients initially will have to be on a standard care therapy of beta blocker and or flecainide. They have to have had a history of arrhythmia, ventricular arrhythmia, be negative for the AEV antibodies. But before we go on, I'll want to just go back to the history of arrhythmia and be able to perform a treadmill exercise test. The last one, able to perform a treadmill exercise test, is because as part of the study in a highly monitored setting, what we're going to do is we're going to be looking at how the therapy, this SGT-501, impacts the exercise of patients with CPVT. And what we're hoping to see is that it makes it even harder for them to have extra heartbeats. When I say history of ventricular arrhythmia, this means that we're looking really for a patient that has had a history at one point of either what we think was a arrhythmia from their CPVT that may have manifested in either a very dangerous ventricular tachycardia or syncope, meaning that the patient may have fainted or felt like they were going to faint and had a documentation of a heart rhythm that is typical for CPVT. So now I want to talk about the patient's journey through the clinical trial. So we will start before the patient is dosed with SGT-501 with what we call a pre-screening period. And this is when we're going to work together with that participant to make sure that we understand all of those aspects I talked about before, what their genotype was, how they came to a diagnosis with CPVT, as well as make sure that that participant doesn't have any other medical condition which may, one, make this experimental therapy potentially have some increased risk, or that could make it very hard for us to understand what might be that other disease versus what could be going on after their dosing. And once we've been able to sit down with the investigator, meaning the doctor that that participant sees, and understand that patient is a really good potential participant in Artemis, we then have the dosing day. On the dosing day, it will be in a hospital setting for observation, given that this is a new therapy. The hospital admission will be over the course of a couple of days, where we'll be able to monitor the patient closely. And then really between day 1 and day 30, there'll be fairly frequent returns back to the clinic to monitor the patient's blood and to check in with the patient to make sure that they're doing well. After that first 30-day period, the patient will be seen relatively infrequently, about on a monthly basis, to make sure they're doing well. But also, as I shared, especially at that 90-day visit, we'll be looking at what happens to the patient's heart rhythms under exercise. And really, it's at that 90-day visit that we'll be looking for that first exercise stress test to make sure we can look for any abnormal heart rhythms. As you can see, there's kind of a gradation in this slide where their dark blue becomes light blue. And what that is attempting to show is that as we go from day 90 to day 180 to day 270, and then that first year, the frequency of the visits do decrease. Once we go into after that first year, we really only have yearly visits to make sure that the patient's doing well from a safety standpoint. And then we'd like to check in on what happens to that, what happens with that the patient's heart rhythm as they exercise. So what will it mean if you were to participate in the Artemis study? So again, because this is a gene therapy, we would want to make sure that we have a commitment to you for at least 5 years of safety follow-up. It does mean that, especially in that peri-dosing period, meaning around the time of the dosing, there would be a short hospital stay. After the study procedure, there would be 24 total visits, but the majority of those are really happening in that first 30 to 90 days. After that, I hope that I was able to convey to you that the visits become less frequent. We will work with our contract research organization. It's an organization outside of ourselves that can help manage, hopefully, well, the inconveniences of being a participant in a trial. And what does that mean? That means that of course, we would pay for the travel expenses as well as accommodations during your stay, and hope to support your brave decision to become a study participant. So if you can go to the next slide. Yep, thanks. So what to expect during one of those study visits? It's really to evaluate, again, the safety of the treatment of SGT-501. We also want to ask and follow you closely in terms of how you're feeling, how you're functioning, with the goal understanding if decreasing the risk of having this arrhythmia can have a beneficial impact on your life. And in terms of, you know, I think that the most important thing that I would like to convey is really the most important for us is that your well-being, both from a safety standpoint, from um, any effect of the drug itself, but also how this therapy hopefully can liberate, the CPVT patient from the stress of having this diagnosis. Next slide. So in summary, we are conducting this study in North America. It's for each participant, the duration of the study would be about five years. There'll be that one intense period in terms of the hospitalization around the dosing. There will be, for that participant, in total 24 visits, but most of those visits are during that dosing period and then the following 30 to 90 days. And the drug itself would be a one-time administration of IV SGT-501. So in terms of any additional study information, you can take a picture of the QR code right now if you'd like, but I believe that these slides will be available to you by SADS. Please do contact SADS with any interest in this study. And with that, I'd just like to reiterate my thanks to Marcia, to SADS, to Dr. Priori and the Priori Lab, but most importantly to you for your interest in dialing in today.
Thank you very much, Dr. Brooks and Annie, for this really comprehensive overview. I want to open the floor to questions. Please put them into the chat feature to everyone. I have one that was sent to me privately. And here it is. Dr. Brooks, how long does it take to see if the gene therapy will work or not?
That's a great question. So I think there's a couple of ways we can think about that question. One could be on an individual patient basis in terms of the biologic function of an AAV gene therapy. The other is, does it work overall, or how well does it work overall to address the disease? I think for the first one, from a biologic standpoint, usually we think that gene therapy, kind of when the gene wakes up and is producing protein and having an effect on the cells and then the organ, the heart, we usually think that takes a couple of weeks. And that's why we will really delay that first exercise treadmill stress test to look at how this therapy can hopefully reduce the rhythms during an exercise stress test. That's really not done until 90 days. So that's plenty of time for the medicine to start working, and the medicine here being the gene therapy. It also, Marcia, is the reason why we want patients to be on their background medical therapy, meaning their beta blocker and their flecainide, because we don't know exactly when that time is, and we want to make sure that patients are really well protected. If there's anything I can convey, that's the biggest message, is we want to make sure that patients are well protected. So that's why when we talk about patients coming in, they have to be on a stable dose because they really need to continue that stable dose. We need to see the exercise tests be reassuring before we would peel back, we would even think of the investigator peeling back any of their background medicines.
Okay, thank you. All right, Carrie has quite a few questions here. Thank you, Carrie, for submitting these. She asks, are you hopeful that those with CPVT with an ICD and a history of an SCA will be able to be ICD free and or medication free after therapy? That's her first question.
Absolutely, that's my hope. We are taking a deliberate stepwise approach to the ultimate aim, and that ultimate aim is really to liberate participants or patients, frankly, to liberate participants from hopefully the medicines, the fear of having an event and the need for an ICD, and especially the shocks that can be associated with having an ICD, whether or not those shocks were appropriate. That's absolutely our target. That's why we're committed to the community. That's why we're so excited about this medicine, because we hope that this medicine gets us there. That's our hope. Of course, I can't claim that this medicine will do that, but that is absolutely our aim. So we're starting with a stepwise approach. That approach is to look at safety and initial signs of how the drug is working in this phase 1b study. And as we continue, we hope to move towards the goal of liberating patients from the need for everyday dosing of their oral medicines, and then, of course, the need for the ICD. That is 100% our hope.
Thank you. Going on, would a female still have a 50-50 chance of passing the RYR2 gene to her offspring?
It's a fantastic question. Our medicine, this gene therapy, does not affect the genes in the body itself. So we're not editing the gene. We're not editing RYR2. We are basically enhancing the levels of calsequestrin through this therapy, which allows for the expression of calsequestrin. And that enhanced expression of calsequestrin, we believe, can normalize abnormal RYR2 calcium leak, because the calsequestrin soaks up the extra calcium in what's called the sarcoplasmic reticulum, so that when you're under stress, what we see is less, or what we believe is less leak of that abnormal RYR2 channel and diastole. This therapy does not change the RYR2 gene that is in the body. It is to add extra calsequestrin to then normalize that abnormal RYR2 gene function.
Okay, thank you. And the last part of Carrie's question, what is the immune system suppression entail?
Thank you for asking the question. So we are using steroids as well as a drug called eculizumab, which can interact with some of the downstream effects of the antibodies that are released in the bloodstream after the after the gene therapy, and then a drug called sirolimus, which can also reduce the activity of the T cells and the B cells. B cells make antibodies, T cells make cells that go in and can actually cause that heart inflammation, that liver inflammation that we talked about. And so by a short course of these drugs, steroids, a drug called eculizumab, and this drug called sirolimus, we believe that we can have the best effect to try to suppress that immune response.
Okay, thank you. A related question from Charles, is an immunosuppressant given?
Yes. So exactly. So we have a short course of eculizumab, sirolimus, and prednisone in the first couple of weeks after therapy to suppress the immune system, to try to decrease any risk to the patient. And those risks are kind of what we walked through before. which are known risks to the liver and some heart inflammation.
Okay, thank you. Catherine is a provider and she's curious as to where are the clinical sites that you currently have, or if you have those identified that you can release now or not.
Yes, because I believe that they're on clinicaltrials.gov, which is a wonderful place to go to get more information about this study. And so where we are going now is the Mayo Clinic. There's a site in Vancouver as well. We're also in Cleveland and Philadelphia. I think I have the ones that are on the ClinTrials. If I've forgotten any, I apologize.
All right, thank you. Let's see. Okay. Mickey says, when would the medications be discontinued?
This is a fantastic question, and I will be honest, we don't know. Because we're not sure exactly when we're going to see the optimal levels of the calsequestrin expression. That's why we want to make sure that all of the patients start, all the potential participants start on a baseline of their normal therapies. And then what we want to see is that those patients can do stress tests without, what we're hoping to see is that any risk of arrhythmia, any abnormal heart rhythms can be reduced. And in consultation with that participant's physician, what we call a principal investigator at a site, and by looking at the exercise stress test results, as well as we do have patch monitors to monitor the patient at home. If everything looks reassuring, it's only at that point that we would contemplate, again, in discussion with the participant's physician, that investigator, a reduction in the medicine. And then we would want to see that participant get on a treadmill again. So we make sure they're safe. So that's really the order of events, making sure that participant's doing well from an arrhythmia standpoint. Then we assess with the investigator all of that data. Then we would contemplate a reduction in their dose of medicine, and then we would check again the exercise stress test to make sure that they continue to be as safe as any way that we can investigate that.
Okay, thank you. Stacy asks, is there a minimum number of participants required for the study?
Yeah, so I think what we're looking at are kind of groups of 3. And so initially we'll have the first 3 come in, look at all their data, and then we would decide if we would expand the study. Along with that, we would look at how those participants are doing. So now maybe we're at 6, and it's at that point that we would say, do we have enough data to really start to think about those participants that are younger than 18? And then we would do another group of 3, take a look at them, look at the safety, and go from there.
Great, thank you. Anna asked, will participants stop their current medication during the trial to see if the full effects of the gene therapy, especially in stress tests?
Absolutely. Thank you, Marcia. And absolutely, that is something that we want to approach with a lot of deliberation. That is our goal. I will not hide that at all. Of course, that is our target, is that we can have a gene therapy that can liberate the patient from the need for these drugs, because we know, number one, that if they're not taken, even accidentally, that can be a very dangerous situation for the patient. So we want to be able to be a safety net for that patient. And number 2, those drugs sometimes can make the patients feel sick. So we want to liberate them from those feelings as well. At this point, Marcia, we don't know how long it's going to take SGT-501 to work, or to what degree it works. And so we will need patients to start on their medicines, and then in a very deliberate and careful manner, we'll be looking at all of that data in terms of how they're doing on the stress tests, how they're doing in free living, with the patches. And then only at that point where we feel that the participant looks really good, that we would then talk to the investigator about potentially reducing the level of the medicine, testing again, and seeing how they're doing.
Okay, thank you. Okay, we have a couple questions related to the age group 7 to 18 and when you plan to have that into the study.
Absolutely. So as you can imagine, and the FDA, and we really share this opinion, feel that when we talk about patients who decide to be very brave participants, um, that are younger than 18, we need to approach that with some knowledge, because that is a vulnerable patient population. And so we want to get through, it's really a function of the data we're seeing, but we want to get through at least that first 6 participants before we would contemplate coming back to the FDA. If we feel that everything is going well, to come back to the FDA and say that we have now demonstrated through the participant through 6 brave participants, the safety of SGT-501, are starting to see what we would hope to say is some positive effects on their arrhythmia before we say, we really want to get into the adolescent patient population because we know that's where there's a lot of risk, and that's where we hope we can really make an impact. So I'm not answering your question definitively because we actually have to see the data come in, but it's really that first 3 to look at the initial safety, then expand to 6, really understand what we're looking at before we would contemplate really coming back to the FDA and coming to you to say that we think that this would make sense for an adolescent.
And thank you, Annie, for posting the clinicaltrials.gov site. Here it is again. I went to it. It does have the 3 locations listed: Boston, Cleveland, Philadelphia. It doesn't have Vancouver on here because it's out of the US, I guess. Okay. And Nanette asked when you anticipate expanding trials to other locations, and if so, when?
Yeah, so it's a good question. I think right now those are going to be our trial sites, I think, for at least the next 6 months. And the reason why is because we are following a dosing schedule that really allows us to look at each individual participant as they come through for a set period of time. We want to look at the first 30 days, make sure things are going okay before we would dose the subsequent patient. And if you can think about that, that means that stagger that which is what we call in drug development this the first dosing period, or sentinel dosing. That means that things stretch out. Because things stretch out in that first 6 months, we only have a few sites where we can pay a lot of attention, and we can partner very closely with those sites. So we can't kind of have a far-flung network of sites yet, while we're still really looking at this initial, what we call sentinel dosing period.
Thank you. How long are you required to stay near the clinical trial site after receiving the gene therapy?
This is a great question, and one that this is universal. So for all gene therapies, we really like the patients to kind of stick around for the first week or 2 weeks. And the reason why, there's a logistic reason and then there's a safety reason. So even if the patient's at home, we know it's really in that first week or 2 weeks that we need to follow the patient closely to see if they need any extra medicines. Then the other piece is logistic, which is that patients often, in that first 2-week period, are often going to be at the clinic to have some blood draws or to have some physical exams to make sure that we're monitoring safety appropriately. When I say that the patients might need some extra medicines, what we do find universally with AAV gene therapy is in the first week, patients kind of feel like they have the flu. And so almost universally, patients feel a little nausea. Sometimes there's vomiting. There can be fever. That's kind of what you expect with an AAV gene therapy. That resolves. But as we're starting a new therapy and patients are feeling a little fluish, we kind of want them to be close to the hospital so that we're not only monitoring safety, but if they need anything for, let's say, nausea or for a fever, that site is right there ready.
Very good. Do you work with the patient's current cardiologist during the trial?
So we are what we call a sponsor, which is a drug company making the medicines. One thing that we don't do is we don't contact you in terms of actually working with you in the clinical trial. We partner with physicians that are research physicians. So they're physicians that see patients. They have a clinic, but they also participate in clinical research. Those um, physician scientists are what we call principal investigators. And those are the ones that we work with on what we call a protocol, which is just kind of the recipe of the clinical trial, kind of how things go along in the clinical trial. And that physician scientist is the one that sees the patient who then bravely decides to be a participant. Generally, inevitably, there's going to be a close relationship between that principal investigator, that physician scientist that's actually running the study, and the participant's home clinician, their cardiologist, just to make sure that things are going well. So that's usually how it works. We wouldn't contact the patient's home cardiologist. It would really be the what we call the principal investigator, that physician scientist who's actually conducting kind of the day-to-day research.
Makes sense. Mickey asked about the cost and if it's covered by insurance. I know you covered that Solid Biosciences covers the travel costs and cost to the participant, but if you'd like to review that again, that would be great.
Yeah, and I know Annie is extremely well positioned to be able to respond to this. But I'll start, and Annie, you please correct me. But our goal is to be able to accommodate is really to be able to ensure that any patient that wants to be a partner with us and be a participant in this study, that their needs are met. And we have various mechanisms to do that. Annie, I don't know if you want to.
Yeah, no, I'm happy to add to that, that there is a travel concierge that is contracted as a vendor to the study and will work in partnership with all participants. And so there will be a dedicated resource available 24 hours a day to make sure that all travel bookings, accommodation, that everything is looked after. So there is no cost to participation. There is no claims or anything that will need to go through insurance. And we just try to make sure that we make this experience as smooth and to reduce the burden as much as humanly possible.
Okay, thank you, Annie. LG comments that on the clinicaltrials.gov site, Mayo Clinic is not listed at this point, but they are a participating site, if that's what you said.
That's correct. Yes, absolutely.
Okay.
And just to add on the clinicaltrials.gov, so as and when sites get activated, we will have more contact information that will appear on clinicaltrials.gov. So right now, it's limited information. It just includes a few of the institutions or those hospitals, but we will be adding additional information that will include the name of the principal investigator, the study coordinator, also a telephone number and an email address. So we will be adding information to clinicaltrials.gov.
Very good. All right, that concludes our questions. And I want to thank Dr. Brooks and Annie for their time today in providing this excellent overview of their gene therapy clinical trial for CPVT. And I thank you, the community, for joining us today. So we appreciate your time too. Thank you.
Thank you.
Actually,
Thank you so
Marcia,
much.
looks like there is one more question if we want to sneak one in the last few
One moment
minutes.
here. Let's see what that was. Here it is. From LG. If the trial is successful, how long does it take to get it FDA approved so insurance may cover it?
That's a great question. And let me try to be detailed to honor, I think, what's an important question. There are filing deadlines that the FDA has to make. So when we come to the FDA with a package that details all of the results of all of the clinical trials that we conduct, the FDA has to review that within a certain period of time, and it can vary. Depending on the rarity of the disease and the unmet need. And so here, I think with CPVT, we have a very compelling unmet need, and it is rare. So I think, the time frame we're talking is, between 9 months and a year, after we've conducted everything and we can bring it to the FDA. In terms of insurance reimbursement, one of the things that's incredibly important is that we are partners. We're partners with SADS, we're partners with the community. If we are successful in developing a compelling drug, which is 100% what our focus is, that liberates patients from the need for flecainide and nadolol, and reduces a risk of shock, and maybe hopefully can liberate patients to exercise and get more out of life. That's our goal. If we can do all of that, then I think it's going to be a partnership with everybody here, to make sure that we can come to CMS, which is really the government's insurance company, and other private insurers to make sure that drug can be reimbursed. So that's really the process, to be quite honest. We first have to develop the data. The first step is safety, understanding how this drug is working in patients. Then we can get to what we say is efficacy, meaning proving that the drug really makes an impact on all those things that we talked about. And then once we do that, we will all work together to say, okay, here's a compelling drug. Now we need to get it into the offices of doctors so they can deliver it to patients.
Thank you. And Nanette asked one more question about the FDA approval then is in approximately 6 years, with the trial being 5 years.
That's a great question. No. And that's very insightful. Not necessarily. So we look at 5 years. The 5-year time frame is the safety that we will be following each and every participant. So we're mandated to, and we're happy to do it, to follow those patients for 5 years. Now, that doesn't mean that we have to wait until the end of that long-term follow-up to file with the FDA. I'm really glad that question was raised. We can file much sooner than that, depending on the amount of safety and efficacy data that we've collected to date. And then, we would continue to follow participants to make sure that they remain safe, but that could be after an approval. That's a great question. The overall approval timeline doesn't need to follow necessarily to the end of that long-term follow-up of patients. That's just something that we do because this is gene therapy, to make sure that things continue to be okay with that patient.
So Mickey's final comment. So the idea is this is a lifetime fix if it all works well.
Well, that's our aim. We, Solid Biosciences, are going to be very humble about making any statements other than to say that is certainly our aim. And the first step for us is really to look at safety here. And then the next step is, how is this drug affecting patients and their and under exercise and other living. And then we can really start to investigate, is this a lifetime fix?
So excellent. All right. That's our final comment and question. So thank you very much, Dr. Brooks and Annie, for your time. And we appreciate your support of this, providing this information to our CPVT community.
Thank you so much, Marcia.
Have a good day, everyone.
Thank you. Thanks.